A bacteriophage enters a bacterium with two very different possible futures. It can turn the cell into a phage factory and burst it open, or it can tuck its DNA into the bacterial genome and wait. The tension is that this looks like a choice, but it is really a molecular switch shaped by genes, cell conditions, infection context, and chance. So let's slow down and remove the idea of intention. A phage is not weighing options. After infection, its genetic program begins running inside a living bacterial cell, where regulators, host machinery, and stress signals all influence which pathway becomes stable enough to take over. After a bacteriophage infects a bacterium, two outcomes are possible: immediate phage production with cell lysis, or integration and quiet persistence as a prophage. In the lytic cycle, the phage genome takes over the bacterial machinery, makes many new phage parts, assembles them, and breaks the cell open. That breaking step is cell lysis, like a crowded room suddenly losing its walls. In the lysogenic cycle, a temperate phage follows a slower path. Its DNA joins the bacterial chromosome and becomes a prophage, mostly silent, while the bacterium grows and copies its own DNA. So one infected cell can either burst soon or carry hidden viral instructions into its descendants. Next, we look at what enters the cell and begins directing that choice. A bacteriophage has no brain, intention, or awareness. Its DNA carries instructions that interact with the infected bacterium's chemistry. When that DNA enters the cell, bacterial enzymes copy selected phage genes into RNA messages, a process called transcription. Those messages direct the production of regulatory proteins, which are small molecular switches in a molecular control system. They bind particular DNA sites and change which genes stay active, much like a thermostat changes heating without understanding temperature. One regulator can favor rapid viral copying, while another can keep lysis genes quiet as integration begins. The result is uncertain, because molecule numbers and host conditions vary. So, next we follow the early race between regulators. Early after infection, different phage regulatory proteins begin pushing the same DNA toward different genetic programs. Some early proteins help turn on lytic genes, the genes that copy phage parts and prepare the bacterium to burst. Other proteins do the opposite. They repress those lytic genes and help stabilize lysogeny, where phage DNA stays quiet inside the host genome. A useful picture is a gene control diagram with two teams arriving at switches. One team opens access to lytic genes. The other locks those switches down. The outcome depends on balance, timing, and thresholds, not intention. If lytic activators accumulate first or strongly enough, lysis dominates. If repressors build and hold, lysogeny becomes stable. Next, the host cell's condition explains why that balance shifts. The infected bacterium is not a passive container. Its health and resources affect the phage program. In a fast-growing, nutrient-rich cell, RNA polymerase can transcribe phage genes efficiently. Ribosomes are abundant. DNA replication is active, and host factors the phage relies on may be readily available. That can raise or stabilize certain phage regulators and make production of new viral genomes and particles more feasible. In a stressed, slow-growing cell, transcription may slow, proteins may be degraded or folded differently, replication capacity may be limited, and key host factors may be scarce. The same phage genome can therefore face a different regulatory balance in each cell. So next, we shift from one host cell's condition to signals from the wider infection context. Some bacteriophages are influenced by how many infections are happening in the same cell or nearby population. One cue is multiplicity of infection, meaning the number of phage particles entering one bacterium. If several related phages enter together, their regulatory proteins and genomes can reach higher levels quickly, which can bias the lysis-lysogeny switch toward hiding. A single phage entering a fast-growing cell may face a different balance than many phages entering crowded bacteria at once. Some phages also release small extracellular molecules after infection. As more infected cells appear, this chemical signal builds up outside cells. This is chemical signal, not awareness. Binding to molecules changes regulatory probabilities, not intentions. Once lysogeny is favored, the next question is how hiding is maintained. Once a prophage is integrated, hiding is maintained as a stable genetic state inside the bacterial chromosome. It is not a sleeping creature with intent. It is DNA held under regulation. The prophage makes repressor proteins that bind specific control regions on DNA. When repressors occupy those sites, the lytic genes stay off, so enzymes and shell-building instructions are not produced. During division, the bacterium copies its chromosome the usual way, including the prophage segment. One cell becomes two daughter cells, and each inherits bacterial DNA, with the prophage already built in. This maintenance can persist for many generations, although small molecular fluctuations remain. Next, we look at how severe cellular stress can weaken repression and let the hidden phage wake up. Induction is the molecular shift that can move a prophage from quiet lysogeny back toward lytic growth. It often begins when the bacterial cell is under serious stress, especially when its DNA is damaged. The bacterium activates stress response systems to repair that damage, but those same systems can reduce the effective activity of phage repressors. With fewer working repressors holding the switch closed, lytic genes may begin to turn on. In our example, damaged bacterial DNA triggers a stress response, repressor activity falls, and the phage genes for copying, building, and escaping become active. This is a trigger, not a promise. Some cells may stay lysogenic. That uncertainty leads us to the deeper point. The switch is regulated, but not perfectly deterministic. A phage does not make a simple yes or no choice between hiding and bursting the bacterium open. Its fate is better understood as a regulatory threshold that shifts the probabilities of lysis or lysogeny. That threshold depends on the phage genotype, the host cell's condition, regulator concentrations, and how many phages infect the same cell. Communication molecules, bacterial stress responses, and random molecular noise can all push the system slightly one way or the other. A balanced scale diagram is useful here, but only as an abstraction. There is no tiny scale inside the bacterium. Instead, molecular signals accumulate, compete, and fluctuate until one genetic program becomes more likely to dominate. So, phage fate emerges from molecular regulation inside a changing bacterial cell. By the end, the lysis versus lysogeny outcome should feel less like a tiny organism making a plan and more like a threshold system.